Material and methods. Cell culture. The clonal, embryonic mouse hypothalamic M-hypo E44 and 46 cell lines that were generated under culture conditions. In University of Toronto, Division of Cellular and Molecular Biology, DDB, Toronto General Hospital Research Institute, University Health Network, Toronto, Ontario, Canada M5S188. Before their use in University of Bradford, West Yorkshire, United Kingdom, Richmond Road, BD71DP. Cell lines were cultured in a monolayer T75 flasks and allowed to reach 80% to 100% confluence. Then those cells were plated into 6-well multi-well plates and cultured overnight at 37 degrees Celsius in 95% humidified atmosphere with 5% CO2 in order to enable them to adhere on the well's surface. The next day, all wells were washed with PBS, coated with serum-free low glucose, 25 millimolar glucose, Dulbecco's modified Eagles medium, DMEM, supplemented with 5 milliliter amphotericin B solubilized, fungi ozone, 5 milliliters of 60 lg/ml penicillin, 5 milliliters of 100 lg/ml streptomycin, 5 milliliters of 200 millimolar L-glutamine, and incubated for 6 hours. After this, the media was removed from all wells in vitro. Then the wells washed with PBS in order to remove any floating neuronal cells. And then it was added low glucose, 25 millimolar glucose, Dulbecco's modified Eagles medium, DMEM, supplemented with 5% photal bovine serum, 1000 mg L, 1% amphotericin B solubilized, fungi ozone, 1% of 60 lg/ml penicillin, 1% of 100 lg/ml streptomycin, 1% of 200 millimolar L-glutamine for all wells, except of the 48 hours time point well in which it was added 10 millimolar dilution of Chemerin. For each of the following 24 and 6 hours time points of treatment, the media was removed. Then the wells were washed with PBS, followed by addition of 10 millimolar dilution of Chemerin. And these 6 well plates were incubated overnight at 37 degrees Celsius in 95% humidified atmosphere with 5% CO2. By the end of the experiment, the cells from each well were washed with PBS, trypsinized with 100 L-trypsine EDTA 0.05% phenol red, mixed with PBS, and transferred in 1.5 milliliter microcentrifuge tubes for centrifugation, after which the cell pellet was collected, and then mixed with 100 L of triazole, and stored in the minus 20 degrees Celsius freezer. QRT-PCR, total extracted RNA, was isolated using direct-whole tmRNA mini-prep plus kit, and reverse transcribed to cDNA using BioRat's iScript, trademark, reverse cDNA synthesis kit with MMLV reverse transcriptase in the presence of 2 LML oligo-DT primers per sample as instructed in the manufacturer's protocol. Oligonucleotide primers were used, which are designed to amplify specific regions of the cDNA sample. The following primers were used in order to conduct QRT-PCR and gel electrophoresis, which were provided by Sigma Aldridge. For BDNF were GTGACTGAGTTCCACC forward and GACGTTTACTTCATTCATGGG reverse. For MTRK2 were ACGAGACACCCAATTAC forward and CTTTGTTCGTAGDATCCCC reverse. For NGF were ATAGGTTTGCCAGGAC forward and GACATTGCTATCTGTACG reverse. For NGFR were CACTTCATTCCTGTCTATTG forward and CTGTTCCATTCTGAAGC reverse. For CNTF were CTAGTGAGCAGAGCAAC forward and TTCAGGCCTTGATGTTTAC reverse, and used as a housekeeping gene. B-actin bit were GATGTAGGAGGCTTTGGTC forward and TGTGCACTTTATTGGTCTC reverse. Quantitative RT-PCR analysis was conducted in ECO48 PCR Max machine by loading in the machine's corresponding 48 well plates. In each used well, 1 L of cDNA or distilled water for the negative control wells, and 9 L of the pre-peppered master mix, which contained 4 L of working primers dilution of 972 L distilled water, 14 L forward and 14 L reverse primers of the selected gene at 100 molar concentration, and 5 L of the IDAC-TM universal silver green for each well. The QRT-PCR analysis was conducted in order to verify the amplification of each gene within the linear range after 40 cycles of the following instructed order: at 50 degrees Celsius for 2 minutes, 95 degrees Celsius for 5 minutes, 95 degrees Celsius for 10 seconds, and 60 degrees Celsius for 30 seconds. In addition, the amplified PCR products for each gene was subjected to agarose gel electrophoresis and visualized by UV illumination in the presence of ethadium bromide. The number of the bands, as well their intensity, were evaluated using UV box imaging acquisition and analyzed by gene size software for visualization of the number of bands to assess the presence of primer dimers. Gel electrophoresis was also conducted for evaluation of isolated RNA's integrity before proceeding in further cDNA synthesis. Statistical analysis of the data was completed by using one-way ANOVA analysis of variants with post-hoc using 2-key HSD, honestly significant difference t-test on the SPSS software. Results: they were assessed by QRT-PCR after Chemerin treatment, the expression of CNTF, neurotrophins and their receptors, NGF, BDNF, TRKA, and TRKB. By assessing the CT cycle threshold values, which are inversely proportional to the starting amounts of mRNA which extracted from the hypothalamic M-hypo-E44 neuronal cells. This way, the lowest CT cycle threshold number for each gene represents its highest amplitude of expression. From the data collected from 22nd of January to 15th of February, the changes after 6, 24 and 48 hours of Chemerin treatment in the expression of beta-actin, CNTF, NGF, NGFR, BDNF and NTRK2 were observed. The beta-actin expression, 24 hours after Chemerin treatment, was upregulated, while at 6 hours reached its peak levels of transcription, and in the 48 hours time point was downregulated, by comparison with the control's expression. In the case of BDNF, its transcription was upregulated in all time points, while it reached its peak in the 24 hours time point. In addition, the expression of CNTF was decreased at all time points, in comparison with the control's expression. Moreover, NTRK2 mRNA levels were increased only at the 6 hours time point, while those levels were decreased at 24 and 48 hours time points of Chemerin treatment. Also, the NGF's expression levels were decreased at 48 hours, while they were significantly increased at 6 and 24 hours time points. By comparison with the expression of controls, while it reached its highest levels of transcription in the 24 hours time point. Furthermore, NGFR's transcription was decreased in all time points, with the 6 hours having the lowest amplitude of expression. This experiment's data for all the tested genes is illustrated in Figure 18 below. Figure 18, Gene Expression Analysis by QRT-PCR for the expression of hypothalamic markers of inflammation and neuroprotective response following Chemerin treatment in the mouse hypothalamic NPY expressing neuronal cell lines M-hypo-E44. The total RNA was isolated after 6, 24 and 48 hours of cell Chemerin treatment. Subsequent cDNA synthesis took place, which were used as a template for QRT-PCR with primers specifically designed to amplify specific cDNA sequence for beta-actin, CNTF, NGF, NGFR, BDNF and NTRK2, N equals 3-4. The original mRNA levels were quantified using the CT cycle threshold method. All results shown are reversely proportional to the originally extracted mRNA levels for each gene. Furthermore, all results are relative to those of the corresponding control mRNA levels, expressed as mean plus minus sign SEM, standard error of the means, *P0.05 versus control. Statistical analysis was performed using one-way ANOVA analysis of variance with post-hoc using 2-key HST, honestly significant difference t-test. In addition, it was observed by the data collected from 15th of June to 15th of August that beta-actin's translation was upregulated at 24, 36, and 48 hours after Chemerin treatment, while at 12 hours it reaches its highest levels of expression. By contrast, in the 6 hours time point its transcription was downregulated. By comparison to this of the controls, moreover, NGF's expression was decreased in the 6, 36, and 48 hours time points, while it was upregulated in 24 hours, and reached its highest point of expression at the 12-hour time point. In the case of NGFR, its expression was increased at 12, 24, 48 hours, while it reached its peak at 36 hours. By contrast, its transcription decreased at the 6 hours time point. Also, BDNF's transcription was upregulated in 24 hours while it was at its maximum at 36 hours. By contrast, at 6, 12, and 48 hours time points, BDNF's levels were below the expression levels of controls. Furthermore, the expression of NTRK2s was significantly increased only in 12, 24, and 36 hours, while it reached its peak at the 48-hour time point of Chemerin treatment, as can be seen from the data depicted on Figure 19 below. Figure 19. Gene expression analysis by qRT-PCR for the expression of hypothalamic markers of inflammation and neuroprotective response following Chemerin treatment in the hypothalamic mouse NPY expressing neuronal cell lines MHypoE44. The total RNA was isolated after 6, 12, 24, 36, and 48 hours time points of cell Chemerin treatment. Subsequent cDNA synthesis took place, which were used as a template for qRT-PCR with primers specifically designed to amplify specific cDNA sequence for beta-actin, NGF, NGFR, BDNF, and NTRK2, N=3,4. The original mRNA levels were quantified using the CT cycle threshold method. All results shown are reversely proportional to the originally extracted mRNA levels for each gene. Furthermore, all results are relative to the corresponding control mRNA levels, expressed as mean plus minus sign SEM, standard error of the means, *P0.05 versus control. Statistical analysis was performed using one-way ANOVA analysis of variance with post-hoc using 2-key HST, honestly significant difference t-test.